In the last couple of lectures, we've been examining the work of Gregor Mendel, that remarkable monk who used breeding experiments with garden peas in the Abbey garden to infer fundamental laws that govern how parents transmit heritable traits to their offspring. You'll recall, however, that Mendel's work, which was published in the 1860s, went unnoticed at the time and actually was forgotten for 4 decades. Then, in the early part of the 20th century, a new generation of biologists interested in inheritance began to see some of the same patterns that Mendel had described long before. And this led to the rediscovery of Mendel's work. Now, with this rediscovery, the field of genetics simply exploded. I mean, every biologist in the early 20th century was in one way or another interested in the problem of inheritance. Mendel's basic findings were confirmed, and geneticists began to explore what other mechanisms might underlie inheritance, expanding on what Mendel had already established. Now, one of the most critical ideas to be tested by these early 20th century geneticists was to establish that, in fact, genes are located on chromosomes. Now, why was this important? Well, it was only after this connection between genes and chromosomes was firmly established that Mendel's work could be understood completely. And furthermore, it was only after this connection was established that exceptions to Mendel's work, things that emerged afterwards, that newer geneticists were finding, could be explained. Thus, it was the connection between genes and chromosomes that really allowed the modern field of genetics to be established and flourish throughout the 20th century. Also, you'll remember that establishing that genes are physical entities located on chromosomes was the first clue that began the search for the molecular basis of inheritance that we discussed earlier in the course, and led eventually to Watson and Crick's discovery of the double helix in the 1950s. In today's lecture, what I want to do is to look in a little bit of detail at how this connection between genes and chromosomes was forged. Chromosomes hadn't been described when Mendel lived and worked. It was only later, with the advent of improved microscopes in the second part of the 19th century, that biologists began to describe the structure of cells in some detail. And one of the things they noticed were the formation of dark bodies in the nucleus of cells that would appear just before cells divided. And furthermore, they noticed that as cells were dividing, these dark bodies would follow peculiar and highly stereotypic patterns of movements. Of course, what they were seeing were chromosomes and the movements of these chromosomes in mitosis and meiosis. Now, the significance of the movements of these chromosomes wasn't appreciated at first. But then in the early 20th century, 2 cell biologists independently had an insight. Walter Sutton and Theodore Boveri observed that the highly choreographed movements of chromosomes during meiosis reduced by half the number of chromosomes that would be found in gametes. And then when gametes joined, the total number of chromosomes would come back up to its full complement. They realized that this reduction in chromosome number in gamete formation and its subsequent restoration in zygote formation could explain the patterns of trait transmission that Mendel had described with his laws of segregation and independent assortment. In 1903, Sutton and Boveri both independently published their ideas, which have become generally known as the chromosomal theory of inheritance. In essence, they provided a hypothetical mechanism whereby chromosome movements could completely explain these 2 fundamental principles that Mendel had suggested. Now, I want to give you another term. I have to introduce a couple of terms as we go along. Cell biologists back then would, because they were positing that genes were physically located somehow on chromosomes, would refer to a gene's location as its locus. The plural of that would be loci. And still today, geneticists will refer to genes and genetic loci somewhat equivalently, and I might do that as we go along. Now, the chromosomal theory of inheritance should seem obvious to us at this point in the course, because we already know so much about DNA. We've learned that before. But it wasn't clear then that you could establish this relationship. Specifically, it wasn't clear how, during the early part of the 20th century, you could prove that genes were on chromosomes and thus prove the chromosomal theory of inheritance. Sutton and Bovary had suggested this connection, but it was just a hypothesis. Confirmation of this hypothesis actually can be attributed to one particular scientist and a remarkable lab group that he established, and also to the particular organism this scientist chose to work on. The scientist was Thomas Hunt Morgan, who was an embryologist, a biologist studying patterns of development, working at Columbia University. Like most biologists at the time, Morgan too became interested in mechanisms of inheritance as people began to talk again about Mendel's work. Now, Morgan was particularly interested, as he was studying development, in mutations. And he was interested in how new mutations arose in organisms. Many geneticists at the time had begun working on organisms that had more complex patterns of trait transmission than, for example, the garden peas Mendel worked on, including, for example, small mammals such as guinea pigs and mice, because the way that the fur color patterns of these mammals would be transmitted from parents to offspring sometimes corresponded to what Mendel observed, but also led to a lot of interesting exceptions that these geneticists wanted to understand. So Morgan, when he got interested in genetics, set out to work on the genetics of coat color in mammals. But mammals are expensive. They were back then, they still are now, both to buy and to maintain, and Morgan couldn't actually raise the money to do this work. Literally, Morgan couldn't get a grant. This is the bane of all practicing scientists. Well, actually, Morgan's inability to get funded to work on coat color in mammals was probably one of the most fortunate grant turndowns in the history of science, because it led Morgan, by necessity, to start working on a different model organism, the fruit fly, a small little fly that's scientific name is Drosophila melanogaster, or just commonly known as Drosophila. As it turns out, Drosophila very quickly became, and remains to this day, the single most important model organism used in both classical and molecular studies of genetics. Now, from Morgan's point of view, and still today from our point of view, there are a lot of advantages to working on fruit flies. First of all, they're cheap. I mean, literally, Morgan could put out a pineapple on the ledge of his office in uptown West Side New York City, where Columbia University is, and within a day, he would attract all sorts of fruit flies. I'm sure you see this in your fruit baskets all the time. Fruit flies are also very easy to raise in the laboratory. Morgan kept them actually in old recycled milk bottles. And he put kind of a food mash, a food mixture at the bottom, and within these milk bottles, the flies were happy to breed and reproduce, to reproduce prolifically. Most importantly, and one of the reasons that fruit flies remain such an important model organism today, they have a very short generation time. Adult fruit flies will develop from eggs in only a matter of days. And what this means is that it's possible to observe the results of genetic crosses in a very short period of time. You could do a lot of crosses. You didn't have to wait for those garden peas to grow up over a matter of months. Within a few days, you'd know the answer. There were some serious problems working with fruit flies, though, and one in particular I need to point out, and that is that fruit flies that you collect from the wild don't have obvious phenotypic variants. Remember, the pea plants were wonderful because they had these dichotomous traits, purple flowers and white flowers. But if you put out your pineapple and collect fruit flies, I mean, to a first approximation, they all look the same. Or at least they all look the same if you just look at them, morphologically. That is, the fruit flies didn't offer traits that that Morgan could use in particular established crosses. Now, this seems like it's a major problem. How are you going to understand the genetics of trait transmission if there aren't obvious traits that you can follow in your crosses? But remember that Morgan was interested in mutation. So his first goal, really, when he started working with Drosophila, was to see if and how a mutant phenotype might emerge in a natural wild population. Okay, let me stop and give you just a little more terminology before we go on. This is going to be important for our subsequent discussion. Morgan and his students and the legion of people who have followed him studying fruit flies refer to the phenotypes of these fruit flies in particular ways. They refer to the characteristic that you would observe in a wild fruit fly as being the wild type phenotype. Now, because wild fruit flies don't have a lot of visible variation, it means that basically all fruit flies that you would collect are basically just composed of wild type phenotypes for any particular characteristic you might be interested in. Now, if they observed an unusual phenotype, specifically a phenotype that they thought was due to a mutation, they called it a mutant phenotype. All right, so we have wild type and mutant phenotypes that are what we're going to be looking at when we look at fruit flies. And the assumption here is that the mutant phenotype somehow must be the result of a mutation in an allele for the gene responsible for the trait you're looking at. Now, another detail, and this is just a detail, but we need to sort these things out, that I need to mention is that Morgan and the people who have followed him on fruit flies use a slightly different convention for labeling their alleles. Remember when we were talking about Mendel, we used letters where we would use a capital P to stand for the dominant allele for a trait and the small letter p to, for example, to stand for the recessive trait, in that case, flower color. The way that Morgan designated or labeled essentially the kinds of mutant alleles and genotypes he was working with was by labeling the allele according to the phenotypic characteristic of the mutation, of the mutant phenotype. So let me make this clear with an example of a well-known mutation in Drosophila, which involves the reduction in the size of wings. I mean, these guys are just tiny little flies. They have wings. But one mutation that occurs causes those wings to not develop properly. In fact, the wings are all sort of small and scrunched up. This mutation has been labeled the vestigial wing mutation, or just simply vestigial wings. We would label the allele responsible for this mutation VG, just a couple of letters because that sort of gives us some cognate connection to vestigial wing. Now, the interesting thing is that the mutation in this case, or the alleles in this case, are named after the mutant phenotype, not the wild type phenotype. Okay? The allele for the mutant allele would be referred to as just VG, and the wild type allele for that same gene we would call VG plus, a V and a G with a little cross after it. All right, that's just a detail, but when we look at some crosses, we'll want to keep that in mind. Let me make one last point about mutant phenotypes in Drosophila, and it's this. For any particular mutation that Morgan was studying, we can safely assume that the typical wild-type Drosophila, the one that Morgan would just collect out on his pineapple, is we would assume that that individual is homozygous for the wild-type allele. Right? It would have, for example, if we were interested in the vestigial wing trait, if we just caught a wild-type individual, we would assume it's homozygous for VG plus, VG plus. That would be its genotype for that particular trait. Now, if we observe a mutant, that mutant must have at least one mutant allele, right? For example, in that case, it's got to be at least VG plus VG. But actually, more often than not, the mutant alleles that we find in Drosophila are recessive alleles. So if we find a mutation, if we find the mutant phenotype of vestigial wing fly, then we can be pretty sure that it's homozygous for the recessive mutant allele. In other words, it would be VG-VG for the genotype for that particular trait. Now, there's one elaboration I need to make right here, because we've been talking about these traits in the context of genetic crosses from Mendel and beyond. But I want to go back for a second and remind ourselves what the link between these alleles and traits have to be. Remember, we learned a number of lectures ago that genes are stretches of DNA that code for proteins. So what that must mean is that when we're talking about alleles, in any of these crosses we're talking about, those alleles are different versions of the protein. Now, in the case of Drosophila mutations, chances are good that the mutant allele is simply a dysfunctional allele. That's why it's recessive. So, for example, imagine there must be some protein, and we don't know what it is, we don't need to, but we know it's some protein that is necessary for the normal development of wings. That would be the vestigial wing protein, the protein that's produced by that genetic locus. Wild-type individuals would have two functional copies of that particular gene, two alleles that were either the same or that both worked equally as well. The mutant individuals must have two copies of the mutant allele, and essentially these alleles are versions of the DNA that code for a protein that simply doesn't work. So one simple explanation for a recessive allele is that it's a version of the DNA that does not make a protein that does whatever the job of that protein has to be for the expression of the trait. This works equally well for Mendel's traits. Let's go back just for a second to the purple flower color. Remember that we had purple being dominant over white as flower color. Well, what could that gene actually code for? It has to be some protein. Imagine that gene codes for a protein that is involved in some biochemical pathway that generates a purple pigment that eventually gets sent into the flower. The recessive white allele doesn't have to be a protein that codes for a white pigment. It could just simply be a broken protein in that biochemical pathway, so that if an individual has no copies of the purple allele, in other words, it can't make that protein, it only has the recessive allele, then it simply doesn't make any pigment, and it goes to the default white coloration. I think it's important to keep in mind when we talk about genes that all genes are pieces of DNA that code for proteins. And so we should always keep in mind when we're thinking about the effect of a gene, how is it that a protein, that must be what that gene is coded for, could possibly lead to the expression of a trait. And as we'll see later in the course, the relationship between particular genes and particular traits can be quite complicated indeed. Okay, now let's go back to Thomas Hunt Morgan's work. Morgan was raising fruit flies in the lab, and what he was doing was looking for any mutant phenotypes that might appear spontaneously in his cultures. Morgan also actually attempted to induce mutations. Now, nobody knew about DNA and how it might be affected by mutation back then, but what people did know is that certain agents, chemicals and other kinds of noxious agents, could increase the number of mutations that appear. That makes sense based on what we know about damage to DNA now. So Morgan actually would expose fly cultures to mutagens, trying to increase the probability that some interesting mutations would occur. Well, the story goes that actually Morgan's original work was a bit disappointing. I mean, first he didn't get funded to work on mammals, he had to work on fruit flies, then he wanted to look for mutations for fruit flies, but he didn't find any for quite some time, over a year. Well, one day, an undergraduate working in his lab as a bottle washer, as the kid who was responsible for cleaning out these old milk bottles when he was done and washing them up and getting them set for other cultures of flies, this undergraduate named Calvin Bridges happened to notice that at the bottom of one bottle there was a fly that looked very different from the other flies in the bottle. And specifically, this fly looked different because instead of the normal wild-type red eyes that are characteristic of Drosophila, this fly had white eyes. Well, of course, Morgan wanted to find out what this white-eyed fly was about, and it turned out that the white-eyed fly eventually led Morgan, and as it turns out, Bridges and other people in the lab, to be able to demonstrate that genes, in fact, occur on chromosomes, proving the chromosomal theory of inheritance. Let me explain how they did that. The significance of the white-eyed fly can be understood by looking at what happened when Morgan did kind of Mendelian crosses with it. Now, imagine that Morgan took this white-eyed fly, which happened to be a male, I should point out. That's a very important detail. This white-eyed male, and crossed it with a red-eyed fly, which of course had to be a female. In the F1 of that cross, all of the offspring had red eyes. Well, that's what you would expect if this was a standard dominant Mendelian trait. And, in fact, when Morgan crossed these F1 individuals, again as you would expect from a monohybrid cross, in the F2 generation, Morgan saw the classic 3-to-1 ratio of red-eyed to white-eyed flies. So at first glance, it looked like this white-eyed mutation was just some sort of recessive mutation that worked just like the kinds of genes that Mendel had worked on. But there was an unexpected twist to Morgan's results. Specifically, when you looked more closely at the F2 generation, you saw something that wasn't predicted by Mendel. The only white-eyed individuals you found in the F2 were all males. Not all males had white eyes, but the only white-eyed animals you found happened to be males, and there were no females with white eyes. In other words, the 3-to-1 ratio of red-eyed to white-eyed individuals that you would expect from a monohybrid cross came about in this case from all of the females having red eyes and half of the males having red eyes while the other half had white eyes. Let me make that clear. Let's go back and start counting individuals. In other words, what Morgan found is that if he looked at 100 individuals in the F2 generation, he found 75 of them with red eyes, 25 of them on average roughly with white eyes. But it turns out that he would also find on average 50 females and 50 males. You'd expect this. There'd be as many males as females. All 50 of the females had red eyes, whereas 25 of the males had red eyes, and the other 25 of the males all had white eyes. So in other words, you see a 3-to-1 ratio, but with this peculiar difference that it's due to the males having 50% red eyes and 50% white eyes. Another way to put this is that the expression of the white-eyed phenotype, this mutant phenotype, was somehow linked to the sex of the individual. In other words, we would say that this was a sex-linked trait. And sex linkage like this was not at all predicted by Mendel. And it was this sex linkage that eventually allowed Morgan to use the white-eyed mutation to establish the connection between chromosomes and genes. Let me show you how he did that. Well, the first thing Morgan had to do was to establish that it wasn't just that females, for some completely independent reason, couldn't have white eyes. What he did was, in fact, if he crossed a white-eyed male with if he crossed a female, I should say, from the F1 generation, in other words, a female that was raised as the result of that first cross, with a white-eyed male, he would find that, in fact, about half of the female offspring of that cross would have white eyes. So females could have white eyes. But what this result suggested was that somehow females needed to have 2 copies of the same allele to get white eyes, while as males only needed to have 1 copy. Let me explain how that works. The way I need to explain that is to give you one little bit more bit of background, and this is about the relationship between chromosomes and sex determination. Soon after cell biologists began to describe chromosomes in the late 19th century, they noticed an interesting thing about the chromosomes in diploid organisms. And that was that if you looked at the chromosomes in males and females, they were mostly the same, with one important difference. One of the homologous pairs, just one, in all of the chromosomes that individuals had, would appear different in males than they would in females. We know now that the homologous pair that appears different between males and females are what we call the sex chromosomes. The other chromosomes, by the way, are called the autosomal chromosomes. Sex chromosomes are recognizable in general because one of the 2 homologs is usually a lot smaller than the other. For example, in our own case, if you look at the chromosomes of a female, you find that her sex chromosomes will look pretty much the same, the 2 homologs in a homologous pair. But if you look at the male, you find that the sex chromosomes look a little bit different. We call these, by the way, the X and the Y chromosomes, and that's partly because if you squint a little bit under a microscope, the smaller chromosome looks a little bit like a Y. It's a reduced version of the X chromosome. Now, if you look at the autosomal chromosomes, all of the other chromosomes, they all look pretty much, well, they all look exactly identical. That's how you can actually sort of, by looking at them, assign different chromosomes as members of different homologous pairs. Only the sex chromosomes differ in their appearance. In spite of the difference in their appearance, the sex chromosomes nonetheless act as though they were a homologous pair. Now here's the important take-home message. The sex of an individual corresponds to the combination of sex chromosomes that it possesses. The way this works differs in different organisms, but in flies, as in humans, females have 2 X chromosomes, whereas males have an X and a Y chromosome. Now, the last thing to understand about sex chromosomes is that, in general, the reason that the Y chromosome is smaller than the X chromosome has to do with the fact that it literally carries fewer genes. there are many more genes on the X chromosome. It's much bigger than on the Y chromosome, which has relatively few genes. This means literally that for many of the genes carried on the X chromosome, males can only have ever one copy of those, because the Y chromosome that they have, which is the homologue to that X chromosome, simply doesn't even have that gene on the chromosome. It's a very unusual thing about sex chromosomes that differ from the way autosomes work. What we say, by the way, is that male individuals, XY individuals in the case of flies and humans, are hemizygous. To contrast it with homozygous or heterozygous, they can only possibly have one allele, so we call them hemizygous. Now let's go back and look at Morgan's cross with the white-eyed mutant in this context and see how it connects genes and chromosomes. Morgan's result could be explained if the white-eyed mutant allele occurred on the X chromosome. Females carry 2 copies of this gene because they have 2 X's, while males carry only a single copy. Now, if we start at the beginning of Morgan's cross, all females in the F1 generation expressed the red-eyed phenotype. But we know that they're all heterozygous for this particular genetic locus. Why is that? Because they're female, we know that they have to have 2 X chromosomes. But the only way they can get 2 X chromosomes, because they're always going to get one member of their homologous pair from the father, is if the father gives them an X chromosome. So if we start with a cross between wild-type females, which we assume are homozygous for the dominant wild-type allele, and a male white-eyed fly, which we know is hemizygous for the recessive white-eyed allele, and we cross them, we know that all the females have to have in their X chromosomes one of the wild-type alleles and one of the mutant alleles. Males, on the other hand, are XY, and they have to inherit their Y from their father. So we know that all the males in the F1 generation had to get their X chromosome then from their mother, because they had to get the Y from their father. So the only place they got their X was from their mother. This means that all the males in the F1 generation would have on their X chromosome the wild-type allele and then a Y chromosome. They would be hemizygous for the wild-type allele, whereas all the females in the F1 generation would be heterozygous. They'd have one wild-type allele and one mutant allele. Now let's look what happens when we get to the F2 generation. If we draw a simple Punnett square and ask how could the gametes assort in this or combine in this F2 generation, we see that the females all are going to be getting, or the females are all going to be getting at least one of the wild-type alleles, because they have to get a wild-type allele from the fathers, the F1 fathers, because that's all the F1 fathers have to give them. So every female, which has to have 2 X chromosomes, is going to get one of those X chromosomes from its F1 father. And we know that that X chromosome has to be wild-type. Because the wild-type is dominant, all of the females in the F2 generation will have the dominant red-eyed wild-type phenotype. The males, on the other hand, have to get their Y from their father and the X from their mother. Because the mothers are heterozygous, they're going to produce half of their alleles with the wild-type allele, half of their gametes with the wild-type allele, and half with the mutant white-eyed allele. Now, when those alleles are combined, when those X chromosomes are combined with the Y chromosome from the father, the males are hemizygous, so they'll express whatever their one copy tells them to express, and that means that half of the males will have red eyes and half will have white eyes. Again, the details of working out this cross aren't really what's important here. What's important is the significance of what this cross told us. So let's return to where we began, which was how we could demonstrate that genes must occur on chromosomes. The way that Morgan's work on the sex-linked white-eyed mutant made this connection was because he could literally trace the movements of genes on chromosomes, because the sex of individuals labeled which chromosome they had to have for the sex chromosomes in specific. In other words, he was able to follow the sex chromosomes because they were identified by the phenotypic sex of an individual, the way that it was impossible to follow the particular movements of individual autosomal chromosomes. By being able to associate the movement of the sex chromosomes with the phenotype for these individuals in the F2 generation, Morgan was able to demonstrate that genes must be on chromosomes. Actually, it wasn't Morgan himself who did this alone. Calvin Bridges, the undergraduate who found that white-eyed fly, went on to do a PhD in Morgan's lab. And it was really Bridges' PhD dissertation which conclusively made that link. What we're going to do next time is turn our attention now from these Mendelian genetic problems we've been looking at to see how this kind of variation we've talked about leads us to evolution by examining the work of Charles Darwin, whose theory of natural selection provides the framework on which we can understand modern evolution.